Good morning or good afternoon, depending on when you have biology today. I'm out doing a thing throughout the school district, kind of doing one of those shadow behind the eyes of the student thing today. So I'm getting that fun experience at the middle school. So I am not able to be here with you today, but I wanted to still be able to go over today's lecture with you and have you get some of the notes down. So today what we're going to be doing is we're going to do a short introduction to the different kingdoms of biology. There are two, and then kind of go through the different types of bacteria within those two kingdoms. And then the rest of the class time will be work time working on a worksheet for you guys that kind of reviews some of the material that we're covering today, but then it also gets into some of the material we'll be covering tomorrow and then into next week as well. So that's kind of our plan for today. So to begin today's class period, if you haven't already, please go to Canvas, go to modules, click on today's date, which is 2/15/17, and then Introduction to Bacteria Guided Note Sheet. You should have done this yesterday after you took your quiz on taxonomy, but if you did not, if you were absent or something like that, to access today's notes, you need to go to, once again, Canvas, go to modules. We are in the Unit 7B, Bacteria and Viruses unit, so go there, and then you should see the 2/15/17. I think it's just Bacteria Guided Note Worksheet where you can get those notes in. So please upload those into Notability if you haven't already. All right, so to begin today, we're going to do an introduction on bacteria. Within this unit, we'll be doing a bunch of different things. You'll be researching different types of bacteria, learning not only is bacteria a bad thing, but it's a good thing as well. And then we'll be doing kind of a lab in the back where you'll be growing your own bacteria as well, and we'll be testing the different effects of different antibiotics on those particular bacteria. So here is a petri dish with a bunch of different types of bacteria growing on it, and you'll be doing something similar like this in lab as well. This is a pretty contaminated petri dish that we see here. All right, so to begin, we've talked about the differences between a prokaryote and a eukaryote, and bacteria does fall into our prokaryotic category. Reasons for that, or why it is a prokaryote, is right here. We know that prokaryotes do not have a nucleus or other membrane-bound organelles, unlike our eukaryotes, like what we have. We are eukaryotic. We do have a nucleus in our cells, and we do have membrane-bound organelles. Bacteria do not. Also, bacteria, they're unicellular. So uni, once again, means one. And so they are single-celled organisms. We have two kingdoms of bacteria. The first type is archaea bacteria, and we're going to go into different types of archaea bacteria, and we're going to go into different types of eubacteria as well. But what we'll find in our archaea bacteria is that these bacteria are called extremists. They live in very extreme environments. So maybe they like very hot environments, or very acidic environments, or environments where it's very salty. Things that normal organisms cannot withstand, these types of bacteria can. So because of that, they get their own kingdom, which is the archaea bacteria kingdom. The other kingdom then is the eubacteria. And these are also called the true bacteria. These are the bacteria that we're more familiar with. These are ones that we may grow in class, or the healthy bacteria that help make breads and yeasts and beer and things like that. There are also bacteria that we may come in contact with that could cause us to be sick as well. So these are kind of the normal bacteria. These are the very unique, specialized bacteria. All right, we have three groups of archaea bacteria that we're going to be focusing on that do live in extreme environments. That first type of archaea bacteria is called the methanogens. These guys are very unique in the fact that they can live in areas where there isn't a lot of oxygen, or in an environment where there is no oxygen at all. They do have the ability to also make methane gas as a waste product. So since they aren't going through aerobic respiration, which is the use of oxygen, instead they go through anaerobic respiration, in which they produce some methane gas as their waste of that particular process. They can be found in the guts of certain mammals. They can also be found in wetlands, hydrothermal vents, and then also marine sediments. So areas that can be very extreme as well. An advantage to them also is they may be used to help convert carbon dioxide into methane gas, which may be something that can be used as a natural gas to help us in the future. So I have a little clip kind of showing one of the beneficial parts of these particular types of bacteria and how they may help us in the future.
With a world population of over 7 billion human beings comes a huge demand for more energy. The oil, coal, and natural gas industry supplies this energy, but with an ever-increasing costly carbon emission footprint. However, our plan to use methanogens harvested from the swamp-like environments, such as the edges of lake beds and sides of rivers, are then grown in our carbon dioxide microscrubber, utilizing a low-cost 3D printed porous filter to hold the bacteria. Methanogens then convert the carbon dioxide produced by the oil, coal, and natural gas industries back into methane. Combustion converts the methane back into carbon dioxide to create more fuel and more energy. This achieves carbon neutrality, which lowers greenhouse gases, saves money for industries, and lessens the use of fossil fuels for everyone while increasing energy output. Imagine natural gas as a renewable fuel source that we can produce on demand. Using methanogens today could preserve our world for tomorrow.
All right, so that's just one advantage of using these particular types of bacteria to future benefit us. A second type of bacteria that falls into the Archaeobacteria kingdom is the halophiles. These particular types of bacteria are called extremists because they can live in areas that are very highly concentrated in salt. Okay, so to just kind of give you guys a reference, average salt water or our oceans, they have about 35 grams of salt per liter. But in certain areas like some of the Great Salt Lakes, the Dead Sea, evaporation ponds, their concentration can be up to 80 to 300 grams of salt per liter. So very highly salt concentration versus our average seawater. So these particular types of bacteria can live in those areas. So the Dead Sea, I mean, I don't even think a lot of organisms can live there because of the high amounts of salt concentrations, but these particular bacteria can. If you guys have ever caught like video footage or YouTubed the Dead Sea, you can see that people will actually go there and they can float in the Dead Sea because there is so much salt in that particular body of water that you're able to just float on top of it, which is pretty cool to kind of see. So there is an example here of Rose Lake where we've got some halophiles that do live in Rose Lake, and they help make that particular water red. So I'll click on this so you can kind of see it. There's no music to this at all, but you can see that these are the particular halophiles that live there. They are red in color, and because of that, they are giving off kind of that hint. So here we've got some footage of the bacteria. This is from a light microscope. The lake is pink due to this bacteria that's found in that water. So in this particular area, once again, we've got salt concentrations ranging from 80 to 300 grams per liter of salt, which is causing it to be an extreme environment. So it was really quick, but it was just a short look. I'll show it again of the type of halophils or bacteria you may find in this area. All right, lastly, our last type of bacteria in this particular kingdom are the thermophils. These particular bacteria are extremists because they can live in very, very hot and acidic waters. So they can live in water temperatures of 106 to 252 degrees Fahrenheit. So typically these particular types of bacteria we're going to find in hot springs. They may be found in volcanic sediments or hydrothermal vents, kind of similar to our methanogens that we saw earlier on. So you may find these like in Yellowstone. I've got a video where a woman talks about the types of thermophils found in Yellowstone. So we'll watch that to kind of give you guys a little bit more information on this particular type of bacteria.
Thermophiles are organisms that live in hot temperatures, hotter than what would be considered comfortable for humans. Thermophiles are the only organisms that can survive in the temperatures in hot springs, such as this one. The suite of organisms that live in environments not hospitable to humans are called extremophiles. Those that live in cold temperatures are called psychrophiles. Thermophiles love temperatures about 122 degrees Fahrenheit, and some have been found to live in temperatures above the boiling point of water. Usually enzymes denature at high temperatures. Most thermophiles are microbial, but some multicellular species live in these high temperatures. A common non-bacterial thermophile is blue-green algae, which you will see growing on surfaces of hot springs. The various colors viewed in hot springs are either mineral accumulations or thermophiles with some photosynthetic properties. Since discovery, thermophiles have become extremely important for medical and biotechnology. Their unique enzymes make them irreplaceable in high-temperature reactions like copying DNA. These properties have also advanced the food, clothing, and fiber industries, to name a few. Although human, plants, and wildlife are not considered thermophiles, we all enjoy the warm temperatures of hot springs. From the University of Wyoming Extension, I'm Mae Smith, exploring the nature of Wyoming.
All right, so there you have it, just a little bit more on those particular types of thermophiles and how they can survive those extreme conditions, and even how they're being now used to help us in the future, like they talked about in the clothing industry, the food industry, and things like that. So just to review, there were 3 different types of bacteria that fall into our Archaeobacteria kingdom. They are all extremists because they live in very extreme environments. So there was the methanogens. That's because they can live with little oxygen to no oxygen. There were the halophils, which are the bacteria that can survive in very highly concentrated salt conditions. And then our thermophils, that can live in very, very warm and acidic environments. All right, we are now going to move into our other type of kingdom, which is the eubacteria. In this particular kingdom, we find a couple of different types of bacteria. We find some that are heterotrophs, and then we have some that are autotrophs as well. So eubacteria, they are found everywhere. They are those that are heterotrophs. They do not make their own food, so they need to go and find it. So they must feed on other organisms. And that puts them into either 2 different types of categories. They're either parasitic, So they're parasites and they have to live off of another organism. So they feast off of another organism, or they're called saprophytic. That's another fancy term for decomposers. So they will feed off of dead organisms or waste and kind of break those particular organisms down and use it as their nutrition source. So heterotrophs, again, just remember they are the type of bacteria that cannot make their own food. They need to go and get it somewhere else. On the other hand, there are autotrophs that are bacteria as well. They do make their own nutrients. So these are like our plant-like type of bacteria. A couple of ways that they can do this. They will make their own food by being photosynthetic. So like plants, they kind of photosynthesize. So they contain chlorophyll just like our plants do, and they kind of convert that light energy from the sun into chemical energy that they can use to then grow. So just like plants, okay? So particular types of bacteria that go through this process, there's cyanobacteria. They can either be blue, green, red, yellow, so they come in a variety of different types of colors, and they can be found typically in ponds, streams, or other moist type areas. So autotrophs, they can be photosynthetic. They photosynthesize to get that energy. The other type of autotroph form is called chemosynthetic. And for this particular type, they make energy by breaking down sulfur or nitrogen compounds. And particularly, these types of nitrogen or sulfur are either found in the soil or they can be found in the atmosphere. So these particular types of bacteria, they aren't near sunlight, so light isn't available for photosynthesis to take place, because we know that that is a driving source of photosynthesis. Instead, these particular types of bacteria can be found in the roots. So here's actually a picture of a root. You can see all these little nodules on it. There's bacteria that are in those nodules that go through a process called nitrogen fixation. So what they'll do is they'll take nitrogen from the atmosphere and they will convert it in these little nodules is where they're homed, and they will convert it into a usable type of nitrogen that plants can then use. Okay, so they are converting their chemosynthetic or nitrogen and sulfur into usable forms for particular plants. So there are two ways to just kind of review. Two ways that they can be heterotrophic. They're either parasitic or saprophytic. And then there's two ways that they can be autotrophs. They can either photosynthesize or they can go through this process of chemosynthetic where they're converting nitrogen and sulfur into usable forms for plants. So a very key component for our plants, and also a very key component for our nitrogen cycle as well. All right, so those are our two types of kingdoms, the archaeobacteria and the eubacteria. We're now going to talk a little bit about what bacteria looks like. We'll look at their cell arrangement and then their cell shape, and we'll get into the reaction to staining tomorrow. That's where we will start with class. So for arrangement of bacteria, they can come in a variety of different shapes or groupings, I guess would be a better way of calling it. So if you ever see bacteria in a pair, so there's two of them that are together, we use the prefix diplo. Diplo means that there are just two, they're coupled, they are a pair. Bacteria can also come in long chains. So you can see a chain here, there's another chain here. So it's a long connection of a bunch of bacteria together. If you ever see that, it gets the prefix strepto. Okay, and then our last type of formation or arrangement of bacteria is when it's just clumped together. You see more than just two and you don't see specific chains. If it's just kind of a cluster and a clumping of them, it gets the prefix the staphylo. Okay, so why are we talking about this? Why do we need to know prefix of diplo, strepto, and staphylo? that will come in handy when we're actually naming bacteria, because they will name bacteria based off of its shape and its arrangement. To look at the shapes then, bacteria comes in 3 different types of shapes, and they also have scientific names for those shapes. So if bacteria comes in kind of a rod-like shape, it kind of almost looks like some medicine or like a multivitamin that you would take every day. If it comes in this particular shape, it is called bacillus. So that is the rod shape. That's the scientific name for those rod-shaped bacteria. If it comes in a circle, like we see here, it's called the coccus particular shape. So circular, or our spear-shaped bacteria have the name coccus. And then our last particular type of shape is if it comes in kind of these strings, they say spiral, and if it is that, we call it spirillum or spirilleus. Okay, so sometimes we might look at a particular type of bacteria, and if we see that it's rod-shaped and it's a cluster of them, like what we see here, we would call that Staphylococcus, because it is a cluster of our rod-shaped type of bacteria. So that's kind of how we use the arrangement and the cell shape to come up with the name for that particular type of bacteria. All right, so that finishes up today's notes. The rest of your guided notes that you see, we will be finishing up those tomorrow. So we'll get into grant naming, we'll get into the structure of a bacteria as well. So we will continue with those tomorrow. Today, for the rest of class period, what we would like you to work on is a bacteria review. So this is an assignment. To access it, you need to go to Canvas, go to biology, go to assignments, and it is titled today's date 2/15/17, and it's called Bacteria Review. You need to then put this into your Notability and you can begin to work on it. So it is a 2-page assignment. You should be able to answer a fair amount of at least the first page with today's material and the stuff that we covered, going over those lecture notes. The rest of the assignment might look a little confusing to you because we haven't actually covered that information yet. We will slowly tackle more of that coming tomorrow and then also next week. So this assignment is not due, you'll notice, until Thursday, February 23rd. So that's over a week away. So we're giving you a sufficient amount of time to work on this. That's because it's going to take us some time to get through all the material that this worksheet requires. So it's kind of a worksheet that you slowly tackle as we go over the information. Okay, so for the rest of today, please continue to work on this worksheet. You should be able to get a lot of the first page done. If you want to get ahead, there are textbook page numbers. 500 is where it starts. That's 18.2 Archaeobacteria and Eubacteria, and that goes through page, looks like 511. So you could grab a textbook here in class and you could do a little bit of reading on bacteria as well if you want to get ahead on this worksheet as well. So this won't be due until Thursday, February 23rd. It will be something you'll be submitting on Canvas. All right, so please work diligently for the rest of class, and I will see you guys tomorrow.